Hi, welcome to Critical Thinking for Ecosystem Science. This is Lecture 5, Science and Its Pretenders. I'm Dr. Tom Stolgren. This lecture is loosely based on Schick and Von from your textbook, Chapter 6. But I've added a more personal story in the background. So the basic concepts in Chapter 6 were that science seeks to understand the world by identifying general principles that are both explanatory and predictive. Science is self-correcting and a reliable guide to the truth. We now know that the scientific method, however it's described, clearly relies on careful observations. We induce general hypotheses and possible explanations for what we observe. Then we deduce certain things that must also be true if our hypotheses are correct. And then we test those hypotheses by checking out our deduced implications. This is the standard scientific method by which we gain knowledge. So I thought I'd tell this story by a story of my own. And this is about a hike that began in about 1992 in Rocky Mountain National Park in Colorado. I observed leafy spurge, an invasive plant species, in a wet meadow in what looked like a species-rich area. It had lots of native grasses and forbs in Rocky Mountain National Park. I hypothesized from just that observation and maybe from a few other hikes around Rocky Mountain that plants from other countries, that is, those invasive species, must like the same environments as native species do. Habitats that are high in soil moisture, high in soil nutrients, and with plenty of sunlight and water. The good life. Dun 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 dun. They like the good life, just like the native species. Let's see then if we can test whether native species and species from other countries called exotic species or non-native species or invasive species all have the same patterns in this general meadow. So how would we do that? Well, in this test study, we put out 24 randomly selected plots, fairly large plots. 20 meters by 50 meters was the largest plot size. It's a multi-scale design. It has 10 1-meter squared plots nested within it, two 10-meter squared plots, and then in the center, there's a 100 square meter plot. So whenever we went out to the field, we sampled at 4 spatial scales instead of just the typical 1-meter squared plots that are generally put in. At each site, in the 1-meter squared plots, we looked at the cover and abundance of each species in the plot. And then in the larger plots, we just noted the number of new species added as we increased the spatial scale. Then we also collected some soils information on nitrogen, percent sand, percent silt, and percent clay to get an idea of water holding capacity and nitrogen and carbon in the soils. What we found in this first little study was that high native species and high exotic species seemed to coalesce on the landscape. They were in the same spots, and that high native species cover and high exotic species cover were in the same areas looking down over the meadow. So we had areas of high native species richness and high exotic species richness in the same areas. And the same thing was true of their cover. We also found out that those areas high in both native and exotic species cover were highest in soil percent clay or water holding capacity and also total nitrogen in the soils. So this confirmed our hypothesis that native species and exotic species, species from other countries, like the same general areas in this one meadow. But we needed to do a follow-up study to see how general this generalization was. So we did, and we added hundreds of more plots in Rocky Mountain National Park, and then we also added a bunch of plots in the central grasslands in a bunch of different states. And what we found was, in general, at that large spatial scale with those 1,000 meter squared plots, the more native species there were in a plot, the more exotic species there were in a plot. Whether you were in the grasslands or in the mountains of Colorado, you got the same general pattern. The rich got richer. Native species richness created places for exotic species to be also rich. Now, we also learned that the 1 meter squared plots, the data wasn't so conclusive. You could get positive or negative relationships. But at the larger spatial scales, it was pretty much generally true that the rich got richer. So imagine my surprise to see scientists on the other side of the US come up with an opposite conclusion. In their study plots, 1 meter squared, they found that as species diversity increased, that you had a lower probability of invasion. That's just the opposite of what I was seeing in Rocky Mountain National Park and in other areas of the central grasslands. How could two scientists come up with wildly different results? Well, we looked into their study in a little bit more detail. We found out that most of it all of their results came from one meadow and an abandoned agricultural field in Minnesota. And they had artificially constructed their plant communities. So this is 1 square meter plots, and they either added 2 species, 4 species, 8 species, or 16 species to their 1 meter squared plots. And then they looked at the relationship of new things coming in. And they found out that, yeah, fewer things came in to the plots that had more species. So I started to think, how reliable is this kind of an experiment? Their conclusions, by the way, were much broader than that little field in Minnesota. They said that diverse communities will probably require minimal maintenance and monitoring because they are generally effective at excluding undesirable invaders. This was published in Nature, a very respected scientific journal. But I still question those results. So when you think about the way the two studies are done, with a group of dirty boot botanists on the right there going out and covering the landscape and taking careful observations, versus carefully controlled experiments in one area, which ones of these techniques provides a reliable, consistent, and non-arbitrary representation of the natural world? That's a legitimate question to ask. And can those very limited artificial communities, like the ones on the left, ever provide an accurate, that is, reliable, consistent, and non-arbitrary representation of the world? Great questions to ask. So when we look into how most of those experiments on diversity and invasion are constructed, we find that they use a small number of species, usually 6 or less than 25 species total in the experiment. Meanwhile, natural landscapes may have 500 to 1,000 species, so we're not really sure if those 6 or 25 species that are selected for experiments are similar to those elsewhere in the landscape. The size of the species is also considered. So in most experiments, they're usually small herbs or grasses and grown from seed, whereas in nature you find long-lived perennial species that some have large biomass, like trees and shrubs. In the environmental gradients looked at in most experiments, they're usually limited to one small area, whereas in natural landscapes, they vary in soils and microclimate, hydrology, and usually have a mix of gradients running in every direction. Moisture gradients, elevation gradients, invasion gradients. The types of species that are collected and used for most experiments include convenient seeds, whatever they can get your hands on. And yet in nature, there's a greater variety of native and non-native species in the whole seed pool. In the level of protection, most experiments, especially if you go to that much trouble, are very well protected. You stop grazing, you usually halt fires, and you try to stop the ground-dwelling mammals from ripping up your plots. Whereas in nature, these kind of disturbances are very common, and you get even bigger ones, floods and landscapes at larger scales. So the scale of most typical plant experiments is the neighborhood scale, something really small, close 1 meter squared or smaller. Whereas in nature, there's a variety of scales out there, from the neighborhood scale to the extended neighborhood scale, and then to trees and shrubs and other vegetation types nearby. In the temporal scale considered by most experiments, most of them are very short, Most of the experiments are less than 3 years, somewhere around a PhD study level. Some are longer, and some go on to several 8 and 10 years, but again, in those carefully manipulated, carefully controlled environments. So it's no wonder we might get different answers from a diversity experiment than from a natural landscape. Well, we tested this over and over and had multiple studies which came to the same general conclusion that the rich got richer. The more native species you had in an environment, the more exotic species you had in the environment, as long as you weren't looking in tiny 1 meter squared scales. In fact, if you looked at areas that were even much larger, like say counties in the United States, you find out that the relationship is strong across most of the nation. The more native species you had in the county, the more exotic species you had in the county. Basically, using larger areas, larger plots, or looking in larger areas, you came up with this story just about every time, and many other investigators have found this to be true. The rich got richer. So that initial experiment may be slightly misleading. It may be that the more species you add to a larger area, the more heavily invaded it is. Quite the opposite of the initial claims. And it might be that diverse communities probably require maximum maintenance and monitoring because they are generally ineffective at excluding undesirable invaders. So carefully look at experiments and natural observational studies and look into how each are done before you make up your mind. This is my conclusion about small-scale measurement, though. If I gave each of you a carpenter's level and I sent you out all over the world to take a very careful measurement of the slope of the tarmac, the airport, and we all came back, what would we conclude from our series of very capable and accurate small-scale measurements? We would conclude that the world was flat, which a lot of people believed for a very long time because we had taken a small-scale measurement. We had learned a lot about a little area, but not much about the general area. And even if you were in the 12th and 15th century, you would see a boat sail out of the harbor and find out that the mast would be visible a lot longer than the hull. So a careful observation, even way back then, way back even in 200 BC, might tell you that the world really wasn't flat. Another careful observation, but at a larger spatial scale. And earlier people were also able to see lunar eclipses, for example. And you would see by the lunar eclipse the shape of the Earth, if you were a careful observer, by the round shadows on the moon during the eclipse. I invite you all to read a very fun book called The Flat Earth to see how bad ideas hang around for a really long time. So we have certain criteria for the adequacy of a hypothesis. We have to consider testability. Can it be thoroughly tested? Fruitfulness. Can it predict new phenomena in other areas? Scope. The amount of diverse phenomena that are explained. Simplicity. Does it require the fewest assumptions? And conservatism. Are the results consistent with other information that other scientists are collecting? How does this relate to ecosystem science? Well, if we looked at temperatures across the globe, any set of measurements, you'll definitely come up with this sort of relationship. You'll see that in the 1880s to 1920s, it was rather cool, that it had warmed up a little bit between the 1940s and the 1980s. And since the 1980s, temperature has greatly increased in a lot of areas. Now you could subsample this. You could go out and capture 100 weather stations from around the world and see if those patterns still hold. And I bet they do. That's a pretty good sample size. You could argue that smaller pieces of this don't show an increase, but it's hard to argue against the long-term trend. And with sea ice melting, especially Arctic ice, well, you don't have to be a genius to look at the photos and the satellite imagery from 1980 and compare that to the satellite imagery from 2012. Even with somewhat inaccurate estimates right there on the ground, it's easy to see from space that Arctic ice is declining. When we look at reconstructions of vegetation change or extinction on islands, in this case New Zealand, it's pretty easy to see that you can measure some of these attributes. You can look at vegetation change over time, and you can look at the diversity of animals over time and plot them. Now, those larger flightless birds, the ones that taste like chicken, are gone forever. But we can change things now for the better by perhaps restoring some forests where it's been cleared. My point is that ecosystem science is pretty darn complex. It'll involve issues of scaling up, integrating information from remote sensing down to the ground from those little plots we're able to put in. We can look at long-term change and map things and estimate the rate of change in a lot of different areas. It's extremely complex. There is a great deal of uncertainty in it, but it's a great way to accumulate knowledge over time and help save the world. So I'm inviting you to continue on with your careers in ecosystem science and think about saving the world. Thanks.